This content has been specially prepared for World Peace TV Neuroscience, contributing to a greater understanding of how the brain is nourished, protected, and supported through oxygen, glucose, and cerebrospinal fluid dynamics. The brain is an insatiable organ, relentlessly consuming resources to sustain its high-functioning existence. Although it represents only about 2% of the body's weight, it demands approximately 20% of the body's total glucose supply. Glucose serves as the brain's primary fuel under normal conditions, powering the billions of neurons that communicate through intricate networks. In times of starvation, when glucose is scarce, the brain can adapt by breaking down proteins for energy. But this is a backup mechanism, rather than the preferred method of fueling cognition. Glucose enters the bloodstream through the digestion of dietary carbohydrates and must remain readily available at all times. Unlike muscles or the liver, the brain cannot store glucose for future use. Without an immediate supply, neuronal function deteriorates rapidly. If oxygen or glucose delivery to the brain is interrupted, irreversible brain damage can begin within just 10 minutes. This stark reality underlines the critical importance of immediate intervention during cardiac arrest, where prompt resuscitation can mean the difference between recovery and permanent injury. The body ensures that the brain receives a continuous and abundant supply of oxygen and glucose through a sophisticated network of arteries, most notably through the structure known as the circle of Willis. This arterial circle, located at the base of the brain, is composed of interconnected arteries that create a redundancy in the blood supply. If one part of the circle becomes blocked or narrowed, blood can often reroute through alternate pathways, safeguarding the brain's precious tissue against ischemic events. Major arteries, such as the internal carotid and vertebral arteries, feed into the circle of Willis. From there, blood is distributed through anterior, middle, and posterior cerebral arteries, each nourishing distinct areas of the brain. This network is vital, ensuring every region from the cerebral cortex to the brainstem receives the oxygen and nutrients necessary for survival. Beyond blood flow, the brain is equipped with several other defense mechanisms designed to shield it from harm. The first and most obvious barrier is the skull itself, a rigid, bony enclosure that acts as a physical shield against external trauma. Within the skull, 3 specialized membranes, known as the meninges, add additional layers of protection. The outermost meningeal layer, the dura mater, is tough and fibrous, providing a durable outer coat. Beneath it lies the arachnoid mater, a delicate, web-like membrane that cushions the brain and provides space for cerebrospinal fluid to flow. The innermost layer, the pia mater, clings tightly to the surface of the brain and spinal cord, carrying vital blood vessels that nourish neural tissues. The cerebrospinal fluid itself plays a critical role beyond mere cushioning. Produced within the brain's ventricular system, CSF acts as a buffer, absorbing mechanical shocks that could otherwise damage the delicate brain tissue. It also serves as a medium for transporting nutrients to brain cells and removing metabolic waste. The production of cerebrospinal fluid begins in structures called the choroid plexuses, found within the brain's ventricles. These specialized tissues filter blood plasma to create CSF, which then fills the interconnected ventricles. From the lateral ventricles, the fluid flows into the third ventricle through the interventricular foramina. From there, it moves through the cerebral aqueduct into the fourth ventricle. After passing through the fourth ventricle, CSF circulates around the brain and spinal cord, flowing through the subarachnoid space. This flow cushions the central nervous system, helping to stabilize pressure and protect against injury. Importantly, cerebrospinal fluid is not static. Cerebrospinal fluid is continuously produced and absorbed, renewing itself 4 to 5 times each day. Absorption primarily occurs at specialized structures called arachnoid granulations, which allow CSF to drain back into the venous blood supply. This dynamic flow prevents the buildup of excess pressure within the skull and ensures that the brain remains suspended in a state of near weightlessness, minimizing the risk of damage from impacts or movements. The journey of cerebrospinal fluid is carefully orchestrated. It moves rhythmically, driven by the pulsations of cerebral arteries and respiratory movements. This rhythmic circulation not only supports mechanical protection but also facilitates the distribution of chemical signals and the removal of waste products. It's contributing to the brain's overall homeostasis. It's an essential component of the brain's protective system is the blood-brain barrier. Although not depicted physically in the cerebrospinal fluid's flow pathway, the blood-brain barrier serves as a critical checkpoint, regulating which substances can pass from the bloodstream into the brain. It selectively permits essential nutrients while blocking potentially harmful agents, maintaining the brain's delicate internal environment. Damage to any part of this intricate system, be it a blockage in blood vessels, a breach in the meninges, or a disruption of cerebrospinal fluid flow, can have catastrophic consequences. Conditions such as stroke, meningitis, hydrocephalus, and traumatic brain injury all illustrate the vulnerabilities inherent in even the most well-protected organ. Medical interventions often target these protective systems to preserve or restore brain function. For example, in cases of increased intracranial pressure, surgical procedures may be necessary to drain excess cerebrospinal fluid and relieve pressure on the brain. In vascular conditions like aneurysms or ischemic strokes, restoring or rerouting blood flow can prevent or minimize permanent damage. Modern imaging techniques such as MRI and CT scans allow clinicians to visualize the brain's internal structures and fluids in exquisite detail. By observing the flow of cerebrospinal fluid or mapping the arteries of the circle of Willis, doctors can diagnose and treat neurological conditions with unprecedented accuracy. Research into regenerative medicine and advanced neuroprotective therapies continues to push the boundaries of what is possible. Scientists are exploring methods to repair damaged blood vessels, regenerate neural tissue, and even engineer artificial cerebrospinal fluid substitutes for medical applications. The brain's reliance on a steady supply of oxygen, glucose, NAP, and protective cushioning highlights its dual nature as both incredibly powerful and deeply fragile. Its complexity demands a level of protection unrivaled by any other organ in the body. Understanding these systems not only aids in treating disease, but also inspires awe at the intricate engineering nature has employed to safeguard the seat of human consciousness. In the grand architecture of the body, the brain stands as the crowning achievement, enclosed within a fortress of bone, bathed in life-sustaining fluid, and fed by an elaborate network of arteries. Each layer of protection, each pulse of cerebrospinal fluid, each molecule of glucose delivered to its cells, contributes to the miracle of thought, sensation, and life itself. As we continue to uncover the brain's secrets, we also uncover the profound beauty of our own existence, a testament to resilience, complexity, and the delicate balance that allows us to think, dream, and create. This exploration into the brain's oxygen and glucose supply, cerebrospinal fluid dynamics, and protective mechanisms has been proudly prepared for World Peace TV Neuroscience, committed to promoting a deeper understanding of the wonders within us all.